Thank you. I hope everybody can hear me okay. I'm just blown away by this presentation. I mean, the thanks go to you, Dr. Kaufman, Dr. Duarte, Dr. Craner. This is really a miracle. I mean, as a neuroscientist, to see nerves regrow, especially in the retina, which is the brain. I mean, that's amazing. So it might take me a while to come down here, but anyway, it's a pleasure to be here with all of you. It's as you've just seen, this is an incredible time to be doing FD research. Um, I hope our government is listening. The head of NIH is listening to this presentation, so they realize the import of this work and why we have to, as a society, fund research, because it leads to days like this and miracles like this. So anyway, the great news is not only do we have this exciting ASO development, but we have other great promising therapeutics in the pipeline. And more is always better. And so it's a pleasure. I'm going to introduce, could I have the next slide, please? First of all, before I introduce the speakers, I just want to give a shout out to the wonderful people I work with. First of all, Adrian Gilbert, who couldn't join us in the presentations today, but he's a critical part of our team and a great chemist, knows pharma, and has been really instrumental in helping us get to this day. With his absence, Adam Sachs has stepped in, and Adam's going to tell you about all the exciting developments with Tikkun. And this community is amazing. I mean, Dr. Marini, Dr. Slagenhaupt, Dr. Craner, everybody, Carlos. So anyway, the next presentations, as Dr. Kaufman mentioned, the goal for all of us is to increase levels of the ELP1 or ICAP protein. It has two names, and especially want to increase levels of this protein in neurons. So there's different ways to do that. One is with antisense oligos that you've just heard about. Another way is with CRISPRs. You might have heard of CRISPRs. They're in the news. In general, it's called gene editing, and we have a group now, Dr. Elisabetta Marini, Cristiano Alves, and Dr. Slabenhout, who are got a early career award with courtesy and generosity of the Kopelman Foundation through the Claire and Philip Wexler Research Fund. Thank you so much for your generosity. So Dr. Marini is going to give a presentation on gene editing. Then we are also doing a gene therapy project specifically targeting the retina to increase the survival of neurons in the retina. And Dr. Anil Chakuri has taken over that project. It was started in my lab. But now I retired, and now Dr. Chakuri is leading the effort, and it's also helped in its development with Tikkun Therapeutics. And Dr. Chakuri also received a Young Investigator grant from the Wexler Research Fund and the Kopelman Foundation. And again, thank you so much. We're then going to hear a presentation from Dr. Slagenhout on small splicing molecules. You know, she's been working for a while on these molecules, and they're proving very, very exciting and promising. It's another mechanism for increasing production of a normal mRNA, that exon 20 we want included, as Dr. Craner described. And then lastly, Dr. Miguel Weil from Tel Aviv University has got a different strategy for increasing the function of neurons, and he's going to present. And then finally, we're going to turn this over to Adam Sachs, who's the CEO of Tikkun, and he'll tell you about our efforts at Tikkun to help move all of these therapeutics along. So thank you very much to all of you, and let's all enjoy this day.
Good morning, everyone. It's such a great pleasure today to be here and presenting our work on developing a novel gene editing therapeutic strategy to correct the LP1 splicing defect that is responsible of FD. So as we all know, FD is caused by a specific single change in the LP1 lead, and this change lead to low expression of the LP1, particularly in the brain and in the nervous system. So what we are aiming to do is to develop a specific gene editing approach that by restoring the normal letter in the LP1 gene, we can correct the genetic defect of the disease and restore the normal expression of ELP1, particularly in the brain and in the rest of the nervous system. So our approach is known as cytosine base editing. So base editor are tools that are used to make very precise changes into the DNA. They are so precise they can change just a specific letter in your in the genetic code. And base editor work like a GPS guided pencil. One part of the tool that is known as guide RNA guide the base editor exactly in the spot where you have to make the change, and the other part, base editor, make this small change to fix the single letter in the code that is specifically in this case is wrong in the LP1 gene. So what are the advantages of a gene editing approach for FD? So first of all, this is a one-time treatment. So you do it once and never again. So you can permanently correct the underlying genetic cause of the disease with a single intervention. Another positive aspect of this intervention that you can preserve the natural gene regulation. So by fixing the mutation in its original location, you can ensure that the correct gene is expressed at the natural and healthy level in your body. So these are both very important aspect of why a gene editing approach can be advantageous compared with the current available other therapeutic intervention for FD. So in order to develop a base editor for FD, we have to optimize and test a combination of guide RNA and base editor in order to be very specific and precise in correct the wrong letter that is in the LP1 gene, but do not change the surrounding other letter that are in this gene. So we tested many combinations of both guide RNA and base editor, and we came up with a combination that is highly precise in correcting the letter that is wrong in FD. And then we wanted to see if our approach is able to, in fact, restore the normal function in FD neuron. So what we did, we use AAV2 that are harmless viruses that are used in molecular biology to specifically deliver this tool inside the cells. And then we deliver this in FD sympathetic neuron, and then we look and see if in fact this tool works in neuron. And this graph is telling us that our approach precisely corrects the letter that is wrong in the FD, but does not correct the surrounding letter. And the other important aspect is that we look at the function of the neuron. So control neuron, they normally do not fire much. Their activity is very low. What happens is in FD is that the sympathetic neuron, they tend to be very active compared with a control neuron. And so as you can see, untreated FD neuron tend to have way higher activity compared to a control neuron. But after we edit the ELP1 wrong letter using our gene editing intervention, we are able to kind of restore the normal function. So the function of the neuron go back to the normal control activity. And this is a great news because this means that our tool and our gene editing approach work in FD neurons. As a next step, we wanted to see if in an in vivo system, we are able to successfully deliver our tool and make it go exactly where it needs to go to do the correction. So again, we use viruses that are harmless to deliver the tool into a FD paths, and we injected this intravenous so that the tools could travel throughout the entire body and so reach every single cells. And then we look to see if our gene editing approach was able to restore the normal ELP1 expression. So this is a gel that shows that in a mouse brain that is untreated, normally the expression of the functional ELP1 is very low due to the changes of this specific letter in the ELP1 gene. But after we correct this letter, we are able to increase the expression of the functional ELP1. And this graph shows that we are able to increase significantly the expression of the functional ELP1 gene in the brain. And this is a great news because meaning that our tool is able to reach the brain, correct the change, and restore functional ELP1 in the brain. Similarly, we saw exactly the same thing in the liver. So normally the expression of ELP1 is very low, but after we treat the mice, we restore very high level of functional ELP1 transcript. And another important aspect is that we have to make sure that our strategy is very precise. So we only correct the wrong letter in the ELP1 gene, but we do not edit anywhere else in the genome. So we test our approach and in FD line, in neuron and another line, and we really wanted to make sure that our approach only change the letter that is wrong in ELP1, but everything else is maintained normal. And we were able, very impressed to see that in fact our base editing approach is highly precise, and we did not detect any unwanted change everywhere else in the genome. So right now, what we are working on is to see if our strategy can work in the eyes of our mice. So we are trying to deliver this base editor specifically in the eyes using, again, these harmless viruses. And we want to do this. We use FD mice that have the disease, and they also have eye disease, so they also have retinal degeneration. And by delivering this tool in the eye, we wanted to see if we are successfully in rescuing vision in these mice. And this project is in collaboration with Cristiano Alves, that is an expert on gene editing. And I just wanted to highlight that this has been funded by the National Eye Institute, and also this work has been funded also by the FD Foundation, and we are very grateful for the support from the FD Foundation for this specific work on the eye. Another project that we are working on right now is to see if a combinatorial treatment using both the compound that Sue is developing, and she's going to talk about it later today, and our gene editing approach, if by combining these two strategies, we are obtaining even better result in alleviating the symptom of the disease. And so we are planning to deliver the compound using a specially formulated food for the mice, and we are delivering our gene editing tool by still using these harmless viruses. And we wanted to deliver both the gene editing directly into the brain, but also intravenous, systemically everywhere in the body, to see if, in fact, by combining the gene editing and the compound that Sue is developing, we are able to obtain even better result in alleviating the symptom of the disease. This work has been described in a R01 grant that has been resubmitted to NIH on March and is currently under review. And hopefully we can all back from good news from NIH soon. So in conclusion, I show you that our gene editing approach is very efficient and precise in correcting the splicing changes that is responsible of FD. Our strategy restores normal expression of the ELP1 gene and is really much working in restoring the functional activity in FD sympathetic neuron. Our gene editing treatment rescued the splicing defect in mouse, and this is a great news because meaning that has a strong therapeutic potential. And what we are working right now in the lab is trying to optimize this engineering strategy to improve the efficiency and precision of our gene editing approach. We are trying to design an editing strategy to specifically improve vision and target the eye disease in FD. And right now we are also trying to assess if by combining the compound that Sue is developing with our gene editing approach, we can achieve even better result in alleviating the disease symptom. So with this, I want to thank all of you for listening to me and for your attention. I want specifically thank Cristiano Alves, that is the collaborator and expert in gene editing and is working with me on this project. And also I want to thank Nadia Zedner, that is testing our gene editing approach in neuron, and of course Sue Slognap, that is my mentor, and all the people in the lab. And and again, I want to thank one more time the FD Foundation for supporting this work. Thank you all for your attention.
Hello, everyone. My name is Anil Chakori. I'm a faculty member at Scape and Sci Research Institute of Massa, India. Thank you for giving me the opportunity to present my work today based on the gene augmentation therapy to restore vision loss and familial dystonia. I don't want to go deep into the introduction because the previous speakers actually have did a great job introducing the molecular pathology of the FD. But to briefly recap and set the stage for the future work, FD is caused by splice site mutation in a gene called ALK1, which leads to skipping of exon 20 and thereby leading to significant reduction of ELP1 protein, predominantly in the nervous system. ELP1 is a scaffolding protein which plays a very important role in transcriptional elongation, tRNA modification, and cytoskeletal remodeling. And one of the important aspects of the disease, as we know, is the differential splicing effect of the ELP1 with the splice site mutation, where there is a reduced normal transcript, ELP1 normal transcript in the central nervous system compared to the other tissues, non-neuronal tissues. I probably don't need to explain much, but patients with FD, as we all know, has a complex neurological phenotype because of the striking progressive depletion of sensory and autonomic neurons. the two important aspects that we already talked about and have seen excellent results in the previous presentations is about the sensory gate ataxia due to loss of proprioceptive neurons and optic neuropathy characterized by loss of macular ganglion cells, which leads to the vision loss in FD patients. Horatio actually already mentioned it about the vision loss in FD patient. Despite of their complex neurological phenotype, patients suffer from progressive visual dysfunction, which severely affects their quality of their life, which we already know. So one important clinical parameter to measure, to look at the visual loss in these patients is the retinal nerve fiber layer thickness. As we know, there is selective loss of retinal ganglion cells in FD patients, leading to thinning of retinal nerve fiber layer. In this picture, we can see an OCT image where there is a reduced retinal nerve fiber layer thickness in FD patients. My interest as a retinal biologist is to develop a therapy in which I could rescue the visual loss in FD patients. So for that, we actually first started studying the visual loss in FD patients using mouse models. There are a few mouse models that were already known, and one of those models, and all these models have recapitulated, by the way, the retinal phenotype observed in patients. One of such model that I'm going to talk about today is a retina-specific conditional knockout model, which is developed by Dr. Lefcourt's group to investigate the role of ELP1, specifically in the retina. So these conditional knockout mice actually exhibit progressive loss of retinal ganglion cells with degeneration beginning as early as 1 month, as we can see here in this graph. And there will be, there is actually 50% of loss of total RGCs by 6 months of age, which makes this model interesting to study the rescue of retinal phenotype. Again, Dr. Lefcourt's group has made an incredible effort and has demonstrated that that the gene therapy works to rescue visual loss. One of the earlier findings is that delivery of intravitreal delivery of AAV2 human ELP1 vector has shown to rescue retinal ganglion cell loss in this retina-specific conditional knockout mouse model. However, what remains unclear is that whether this structural rescue translates into a meaningful functional benefit, and we can actually make the vision better in FD patients. Specifically, whether the visual function can also be restored using this gene therapy approach. So this is the project that we are studying, and to study that if this gene therapy approach can actually rescue visual function in FD mice. We have conducted a comprehensive evaluation of retinal function using AAV2 human ELP1 vector, using various electroretinographic behavioral tests to specifically assess visual function and determine whether structural preservation is accompanied by the functional rescue. In this presentation, I'll share some preliminary results of our gene therapy approach at 3 months post-injection of this human ELP1 AAV2 vector. So the preclinical trial that we have made consisted of 2 different groups, control mice, which express the human ELP1 in the retina, and the experimental mice, which are called here as a PAX6 mice, which do not express ELP1 in the retina. And within those groups, we have control groups and experimental groups. 2 experimental groups are mice injected intravitreally with 5.4 E8 human ELP1 vector at a concentration of 5.4 E8 and 2.7 E9. These are the 2 concentrations that were used to check if we can rescue visual function. One of the clinical parameters that Dr. Horatio already explained in his presentation is reduction of retinal nerve fiber layer thickness, which is seen in FD patients and also in mouse models of FD. The measurement of retinal nerve fiber layer thickness here is done by optical coherence tomography, which is a non-invasive method to scan the retina. and measure its thickness. So we have specifically measured the retinal nerve fiber layer thickness in conditional knockout mice and the control mice to see, first of all, if there is any defect in reduction of retinal nerve fiber layer thickness in this mouse model, and to see if our gene therapy into the eye has worked to improve the retinal ganglion cell thickness. So here in this graph, we can see that there is significant reduction of retinal nerve fiber layer in the FT mice, that is conditional knockout mice. And then we were able to rescue this reduction, where we can see significant improvement in retinal ganglion, retinal nerve fiber layer thickness in mice injected with AAV2 human ELP1 at 1E8 concentration and 1E9 concentration. Yeah, and next we wanted to check if the structural preservation of retinal ganglion cells has translated into functional rescue. So we performed pattern electroretinography, which is a non-invasive test that is used to measure electrical activity in the retinal ganglion cells to make sure that the retinal ganglion cells are functional in response to the visual stimulus. So the waveforms are shown here. A positive waveform reflects initial depolarization of retinal ganglion cells, and the inner retina reflects the action potential generation and RGC output. So reduced P1 and N2 amplitude, as you see, there are three waveforms. Reduced P1 and N2 amplitudes indicate impaired RGC function. So here in this graph, we can see that there is significant reduction in the RGC function in FT mice, conditional knockout FT mice, compared to the control mice. And we were able to rescue we were able to see improvement in retinal ganglion cell function with the mice injected with human ELP1 at 5.4 E8 concentration at 3 months of age, or 3 months post-injection. And one of the other parameters to check the retinal ganglion cell function is a photopic negative response, which is again a slow negative wave going that follows a B wave in a light adaptive full field ERG. So it reflects the activity of retinal ganglion cells again, and also their axon. So here we can see that there is significant reduction in retinal ganglion cell function, which is indicated by photopic negative response, which again is improved by injection of human ELP1 at a concentration of 5.4 E8, which indicates that the gene therapy can rescue retinal ganglion cell function in FT mice. The other thing we've also wanted to check is to see if the electrical response of the primary visual cortex is passed on to the visual stimuli. So for that, we have performed visual evoked potentials, which are again electrophysiological recordings that measure the electrical response of the primary visual cortex to this visual stimuli. In this, we can these VFPs are also used to access the integrity and function of retinocortical pathway from the retina to the optic nerve in the visual cortex. As we can see in this picture, we are measuring with the visual evoked potential. We are actually measuring this the visual pathway. In the graph, we can see that, first of all, there is significant reduction in the visual evoked potentials, which we measured by flash VEP at 3 months of age in FT mice compared to the control mice. And we've also seen that there is improvement in these visual evoked potentials when the mice are injected intravitreally with human ELP1 vector at 5.48 concentration. This indicates that, in fact, we were able to rescue retinal ganglion cell function and also visual function in FT mice treated with human ELP1 vector. We also performed full field ERGs, electroretinographies, to further investigate photoreceptor dysfunction. Photoreceptors are the cells which are on the back of the eye, which are important for receiving the visual stimulus. And there are two different types of photoreceptors: rod photoreceptors and cone photoreceptors. So full field ERG is a method where we measure the electrical responses from major cell types of the retina, which is majorly dominated by photoreceptor responses, both rod and cone responses, based on the dark adaptation and light adaptation. to further investigate if there is any photoreceptor dysfunction in this mouse model, and if there is any, whether we could rescue these defects using a gene therapy approach, we performed full-field ERG. So there are two different waveforms that we can see here. A wave, which actually originates from the photoreceptor hyperpolarization, both from rods and cones. But if it is primarily coming from the rods, it is from the dark adapted conditions, and from the cones under the light adapted conditions. And the B wave is actually generated by the bipolar cells under the influence of Mueller glia. So here in this, in the too many graphs in this slide, but I'll try to simplify this. So the upper panel are the control mice, in which we should not expect any differences in the electroretinographic responses, where we can see that there is no difference between the control mice treated with a human ELP1 vector compared with the untreated mice. The lower panel indicates the experimental mice. Those are FD mice treated with two different concentrations of human ELP1 vector. The red line here, the red dotted line, indicates the mice treated with the 5.4 E8 concentration of human ELP1 vector intravitreally. We can see that there is some difference in dark adapted A wave amplitude in mice treated with human ELP1 at 5.4 E8 concentration. But we can see that the larger difference is seen in light adapted B wave when the mice are injected with 5.4 E8 concentration, which indicates that, in fact, there is a difference between FD and control mice, and we were able to rescue the electroretinographic responses in these mice. Finally, in order to see if the mice, if we can improve visual acuity and contrast sensitivity in the mice, we performed an assay called as optomotor response assay, which is again a non-invasive behavioral test used to assess visual acuity and contrast sensitivity in these mice. It actually uses a natural reflex which the mice has when they see the moving vertical stripes. So the mice is put on the pedestal, and then we have monitors around the mice with these moving gratings, which are nothing but the black and white striped lines. So the mice will instinctively turn their heads in the direction of the movement to stabilize their visual field. So this the equipment actually records the head movements of these mice, and then give us the values which are known as degrees per cycle. That means that the white and black white paired cycles that can fit in 1 degree of visual angle. So basically what we can see here is a graph indicating the difference between the control mice, that is PAX6G negative mice, and the PAX6G positive mice, which indicates that there is significant reduction in visual acuity in the PAX6G positive mice, which are FD mice. And then we can also see that there is significant improvement in visual acuity in mice injected with both 5.4 E8 concentration of ELP1 vector and 2.7 E9 concentration of ELP1 vector. Similarly, we can also see that there is significant improvement in contrast sensitivity. First of all, there is a significant difference in contrast sensitivity. FD mice showed reduced contrast sensitivity, and then we were able to improve that reduction in contrast sensitivity by injection of human ELP1 vector at human ELP1 vector at 5.48 concentration. So we can see this contrast sensitivity improvement at both two different cycles per degree amplitudes. And we can see that the significant improvement is with 5.4 E8 concentration of human ELP1. To conclude my presentation, our results indicate that AAV2 human ELP1 administration of AAV2 human ELP1 intravitreally in conditional knockout mice has led to improvement of visual function in mouse model of familial dystonia. And we still are in the process of demonstrating the safety and efficacy of this AAV-mediated gene supplementation approach, and we wanted to test the safety of this vector in non-human primates in order to move forward with human administration. And in parallel, we are also doing transcriptomic profiling of retinas treated with human ELP1 to detect the molecular pathways which are dysregulated in FD retina, and also to see if our gene therapy approach can rescue those dysregulated pathways to drive selective retinal ganglion cell regeneration or renewal in these mice. Parallel, we are also working with other groups to generate an iPSC-derived RGC model to actually further investigate if we can test this human gene therapy approach in a humanized model of a humanized RGC model and see if we can rescue these defects that we have seen in mouse models. With that, I would like to thank my mentors, Sue Slogenhaft, Elizabeth Amorini, Daddy, and KK from MGH. And all this work is done by visiting a scientist from Taiwan, Hui-Chan Chang, from my lab at Grossbeck Gene Therapy Center. I would also like to thank Ocular Genomic Institute and my funding sources, National Eye Institute, and more importantly, Familial Dystonia Foundation for the Early Career Investigator Award, which allowed me to perform all this work. Thank you very much.
Hi, everyone. I'm sorry I can't be with you today live on Zoom, but I'm happy to send this recording to give you a brief update on our work to develop a drug to treat familial dysautonomia. I'm Sue Slogenhaft, for those of you who don't know me. I am at the Mass General Hospital in Boston. I'm also a professor of neurology at Harvard Medical School, and my lab is in the Center for Genomic Medicine at MGH. So we've talked for many, many years about correcting ELP1 splicing using a drug. ELP1 is the gene that is defective in FD. And the goal of our work, as you know, is to create a drug that can help us include exon 20. So just as a reminder, in FD, exon 19 should join to exon 20, should join to exon 21. What happens when the FD mutation, which is right here, is present, exon 20 is skipped, and this leads to no ELP1 protein being produced. What we're trying to do is come up with a drug that will correct this such that it goes this way, includes exon 20, and creates functional ELP1 protein. And we want to increase the level of functional ELP1 protein. So that's the goal of the work that we've been doing. So what have we been doing lately? We have a drug called PTC680. And we were awarded an NIH Blueprint Neurotherapeutics grant that I told you about last year in 2023. This work actually began with kinetin way back in 2004, 2005 when we found kinetin. And we've been working since then to create a better drug for FD patients. So we began work with the Blueprint project at the National Institutes of Health in 2012 originally. Worked with PTC Therapeutics for several years, and now we're back working with the NIH Blueprint Neurotherapeutics Network. So this is just a list of the individuals. This is a true team effort coming together to treat FD. My team at MGH, along with those at the FD Center, Dr. Kaufman and Dr. Gonzalez Duarte, as well as many consultants and people from the NIH who are helping us to bring this drug to FD patients. So last year, I told you about the first year of the grant. This year, I'm happy to tell you we've had many accomplishments. The first is we've completed the studies on the pharmacology in mice. We've been able to show that this drug, PTC680, also called BPN 36964, rescues disease phenotypes in mice. So this drug corrects the phenotypes that we see in our FD mice. We've completed studies. DRF stands for dose range finding studies in dogs and in non-human primates. We've completed those studies. We certainly have collaborations established with clinical specialists at the FD Center, so that milestone is complete. We know how to synthesize this drug and that's complete. And we've done pre-formulation screening. So these are all steps in the drug development process. And the thing left to do for this year was to prepare material for our IND enabling studies. And I'll talk a little bit about that on the next slide. The important takeaway here is that all of the data that we've generated strongly support the continued development of this drug as a treatment for FD. So next year, what are we up to? We just got our grant renewed. That is great news that the NIH renewed our grant. And the goal for this year is to do two things: synthesize the drug so we can do toxicology studies. So we need a lot of the drug. And then we need to actually do the toxicology studies, which will give us crucial information about the safety and toxicity of the drug before it goes to people. Okay, so what is happening right now? Nothing. And the reason nothing is happening is because of what is going on with the current administration. More than 800 existing NIH research grants have been terminated. Grant reviews have been held up and research funds have been frozen. So I told you that our grant was renewed. Good news. However, in order to do those two things I just told you about, NIH has to contract with a company in order to produce the drug and then in order to do the toxicology studies. Those are contracts. Contracting at NIH is halted. So our project cannot move forward in any way if contracts can't be awarded. So what can you do to help us? I hope you've already been doing this, but write and call Congress. Tell your friends to do it too. Talk to anyone who will listen. If we can't get funds through NIH that support biomedical research and we can't unclog the contracting, our project will not move forward. So I'm sorry to be the bearer of both good news in terms of the progress we've made, but also bad news in terms of the fact that while some grants are getting renewed, like ours, which is good, without contracts, this project that I just described to you can't move forward. So please help, do what you can. We need to be screaming louder about this, and we need Congress to be screaming louder about this. Lives are at stake. Again, I'm sorry I couldn't be with you live to answer questions. Anil is here with you today. Elisabetta is here with you today. And this is the team at Mass General who has worked on FD for a very long time. And we are determined to keep going. We are determined in order to get this done. We just need a little help from Congress, from the administration, and from NIH to get the contracts going. Thank you. Enjoy the rest of the day.
Good evening, everyone. First of all, I must say how impressed and incredibly encouraged I am with the Amlorem data, the ASO data. To date, a veteran of the industry, I know how much work it takes both in the lab and in the clinic to get to this point. Moved to tears, just heartfelt congratulations. As the father of a 31-year-old FD young man, I guess the burning question is whether these fantastic results will translate to our older patient population, and I look forward to answering that question. My son suffers from severe gait and vision loss issues, and can't wait to see what the future holds. Next slide, please. So just as a reminder, we kind of introduced Tikkun at our last FD Day. Tikkun is a public benefit corporation that we set up as a wholly owned subsidiary of the foundation, the FD Foundation. Its mission is very clear, sort of the business side of things, to aggressively drive drug development for FD, which currently has no available drug treatments, and as its unwavering focus, working with transparency with all of our stakeholders to ensure unfettered access to whatever approved drugs become available for FD patients across the world. You heard a lot about the ASO progress. Tikkun's focus over the last year or so has been our gene therapy vector that Anil spoke about and the small molecule that Sue just spoke about. I want to thank the team that I'm working with, and in particular, Frances Lefcourt, who's been my partner in all of this, could not have made anywhere near the progress without her incredible contribution. Next slide. So the message, and I kind of am echoing Sue Slagenhoff's message here, is that we're at a critical juncture right now. We have never had more compelling data for both the gene therapy vector and the small molecule drug candidates. It's just screaming for more investment. It's screaming for more attention. But at the same time, our geopolitical situation right now, the NIH funding, Sue talked about the Blueprint grant. Recently, we filed an SBIR grant for the gene therapy vector, which is a small business innovation research grant, a grant that's only available to companies like Tikkun, not nonprofits. This funding is at severe risk due to the current geopolitical situation. So we're in a tough spot. We have the greatest data we've ever had and the biggest threat to our ongoing funding that we've ever had. In terms of our progress over the last year, since the last time I spoke to you, we have filed our ELP1 gene therapy US patent application, done. We have executed a gene therapy vector license agreement with Montana State University. It's been completed. About a month ago, we submitted our SBIR phase 1 grant application. That's for the gene therapy vector that was submitted. And just to give you a sense of scale, that phase 1 grant's worth about $300,000, something like that. That's enough to produce the vector for toxicology. And that's when we have to get a new infusion of funds. So it's all about money at this point to keep these programs going. The MGH, Mass General Tikkun sublicense agreement is in progress. We're working hard on that. That's for the small molecule. And I've developed, with some big contributions from our SAB, a pitch deck to be used for both investor and philanthropic community presentations. That's been completed. A couple other quick updates. We've expanded the scientific advisory board for Tikkun to include my good friend, Dr. Todd Wolf, who's the executive director of technology ventures at Beth Israel Deaconess Med Center. And a shout out to Todd, whose birthday is today. And also, most importantly, and this is very timely, we have to get in front of the NIH in what's called an Interact meeting. This Interact meeting is really important. It's informal, and it allows us to have a conversation before we do any more submittals. Are we making the best choices for efficiency's sake, financially? Are we choosing the right animal model for our toxicology? Those kind of questions. I'm very optimistic they will grant us that meeting, and we hope to have that this summer. So, priorities for the remainder of 2025, very clear for us. Protect our IP, continue to get these agreements in place, secure required funds in any way possible. And of course, as you can tell from all this incredible inertia that we have, all this momentum that we have right now, to maintain that momentum with the greatest sense of urgency. So that's a quick update. And I'm now going to turn it over to Dr. Patricio Millar, who's going to facilitate our Q&A. Thank you.